Thank you so much for having me. My name is Joanne. I am from the University of Auckland School of Pharmacy. So I'm going to take the next little while to talk to you about one of the clinical applications of whole brain MRSI. I'm particularly interested in neuroinflammation. So I have, oh my gosh, I should just use this tool. I have no disclosures today. I'd like to begin by acknowledging the traditional owners of the land on which we meet today. I would also like to pay my respect to the elders past and present. So inflammation, just going to give you a really quick rundown. So it's basically, you probably already know this, you've probably already felt it at some point in your life, refers to your body's process of fighting things that harm it. So if your body detects something, an intruder, it launches a complex biological response. So like you get a splinter in your finger or something, a bacteria, a virus, a COVID, a flu, anything like that, your immune system will create inflammation to protect itself and heal itself. So 5 signs of inflammation: pain, you know, you sprained your ankle, I hurt my elbow, it really hurts. It's a result of neuroinflammatory chemicals in the area that stimulate the nerve endings to make it more sensitive. You get heat in the area, it might feel warm because more blood is going to this area to carry immune cells to where the healing needs to happen. You'll probably feel a bit of, maybe also see a bit of redness, which is again related to increased blood flow. There might be swelling, so inflammatory chemicals that cause fluid to leak and build up in that tissue, and you might get loss of function. So this is your body's way of protecting you to tell you to stop, don't cause any further damage. So you might not be able to move your joint properly. Not as intuitive, but like you get a worse sense of smell, you want to stay in bed when you're sick, that kind of thing. You don't always get all the symptoms every time, but you might get a mixture of those. And acute inflammation is there. It's usually short-lived, resolves in 2 weeks or so, depending on what kind of injury you have, and it's basically your body's way of telling you to stop and try and get better. Ooh. Oops. You know, in life people say there's too much of a good thing. My mouse is moving. That's right. Thank you. Too much of a good thing applies to a lot of your life and also applies to inflammation as well. So chronic inflammation happens when you get this, you know, your inflammatory response gets turned up appropriately maybe at first, but then it can kind of continue where it's not as appropriate anymore. It leaves your body in a state of alert. So your immune system is on, keeps pumping out white blood cells, chemicals, so your body thinks it's under attack constantly, and it can last for months to years. So it can result from like your body not or failing to resolve an infection or an intruder. It can be long-term exposure to irritants, so like chemicals or polluted air. It could be an autoimmune disorder where your body mistakenly attacks its own self or its own cells. Obviously, not all of these things will cause chronic inflammation in everybody, and sometimes we don't actually know what causes it. So if you have chronic inflammation, your body's response eventually starts damaging your healthy cells. So it's not helpful. It becomes, you know, harmful to you, and it can, you know, lead to all sorts of sort of DNA damage, tissue death, scarring, and a lot of conditions, really common conditions, are linked to inflammation, like diabetes, heart disease, joint disease, allergies, asthma, but we are at human brain mapping, so what about your brain? That is what I'm going to talk about for the rest of my talk. What about inflammation in the brain, right? How do we measure it? What are we going to do about it? So with animals and dead people, you can get in there, you can have a good poke around and have a look at the cells, right? But people who are alive don't really like it when you do that. So we need to find another way of getting at the brain in people. So you can use measure inflammatory markers in the cerebral spinal fluid by a lumbar puncture. It doesn't really tell you where the inflammation is happening in your brain because you're just getting a little bit of brain fluid from the spine. It's also pretty invasive and unpleasant if you've ever had one. You can do positron emission tomography or PET, as Federico mentioned, radioligands, you know, ionizing radiation. Not everyone has access to it, and it's also very expensive. So, you know, what about MRI? Everyone here loves MRI, obviously, or have access to MRI, relatively less expensive than PET, and so we think that it might be a promising tool to investigate neuroinflammation. So we've talked about sort of the metabolites that you can get, which are really cool. We're going to talk to you, I guess, about one application, or further application of MRSI data by calculating brain temperature. So we thought, it's based on investigating microglial activation, which is accompanied by, your immune cells being really much more active in the brain when the inflammation is present, and upregulation of the surface molecules and production of inflammatory chemicals. So how does it work? So your brain protons exhibit a temperature-dependent chemical shift, but remain stable with respect to things like pH. So there's actually an almost linear dependence of the water proton frequency in sort of from 0 to 40 degrees, which is sort of, physiologically healthy, although, you can get a bit higher than 40 degrees, so that the resonance frequency of water will shift closer to a temperature-independent metabolite by 0.01 parts per million per degree Celsius. Doesn't sound like a lot, but it is, you can pick up a difference as you acquire the spectra. Then you can calculate the temperature based on the function of the chemical shift relative to a reference peak. We like to use creatinine. You can also use NAA and choline, but we find that creatinine stays stable to other factors such as pH, and also is less sensitive to like magnetic susceptibility changes at the cellular level, because you can get sort of differences depending on whether, where there's high concentrations of white matter. The slide I'll just run through really quickly, as it's been touched on already, because you can get obviously the standard or the common metabolites, but you can do it across the whole brain, and you can do it in a much more, I guess, clinically acceptable time than if you were to take all of these single Voxel sequentially. So a lot of the studies that have been done in neuroinflammation have been limited by the use of single voxel spectroscopy. So you can only capture one or two or a few regions of the brain, and you usually have to have a really good idea of where to look. So MRSI, in conjunction with the 3D echoplanar spectroscopic imaging, or EPSI sequence, enables us to get lots and lots of voxels, spectra in lots and lots of voxels, and provide regional temperature information across the brain, in a clinically acceptable time. So we're thinking about 17 minutes in the physicists, I'm not a physicist, I'm a pharmacist, a scan that might normally take about 9 hours if you were to get the same amount of data can be done in 17 minutes or so. And then you can calculate whole brain temperature by averaging spectra across regions of interest or across the whole brain. So it doesn't require us to have an a priori hypothesis. We're just going to look at the whole brain and see what's going on, rather than just having, one or two voxels. So EPSI simultaneously, this is where it saves the time, it simultaneously acquires the metabolic spectra in their spatial location together. And that's how the scans, but don't ask me about the physics of it. So what we did, in order to see if the scan actually works to detect inflammation, is we did a bit of, I guess, a feasibility study of using EPSI to detect low levels of brain inflammation, with a view to applying it to psychiatric disorders such as major depressive disorder, which have, we think, potentially low levels of brain inflammation, as opposed to other conditions such as traumatic brain injury or stroke or multiple sclerosis or meningitis, where you get like really high levels of brain inflammation. So we used a typhoid vaccine, which contained 25 micrograms of polysaccharide from like Salmonella typhi to a healthy volunteer to induce a safe level of neuroinflammation, temporary, short term. We did this, we scanned them before and then 3 hours after giving them this injection, and we hypothesized that following the typhoid vaccine, but not placebo, that participants would show higher brain temperatures across the whole brain and potentially in regions that we think would be associated with neuroinflammation. And we also expected to see some metabolite alterations potentially and across those brain regions as well. So we recruited a bunch of healthy volunteers. You'd be surprised how many people actually don't mind you causing a bit of brain inflammation, especially uni students. So online advertisement, social media, word of mouth. We cut the age off at 55 because older adults can have, they can have exaggerated immune responses to these kinds of things compared to younger individuals. And we took 20 participants who were generally healthy. I'm not going to read through them all, but the main ones really were that they weren't already taking any anti-inflammatory medication, so none of your NSAIDs like ibuprofen and stuff, and that they hadn't recently had any vaccine within the last month, which was quite hard because we started at like flu vaccine season, so it's hard to find people who hadn't already had their flu shot and someone who hadn't had the typhoid vaccine within the last 3 years. So just a quick sort of flow through the study. Participants obviously gave consent. They attended 2 study visits. It was a randomized, it was double-blind, placebo-controlled, and crossed over. So they either were assigned to receive placebo first, then vaccine, or a vaccine first, then placebo, to 2 sessions that were at least 7 days apart, just to allow for a bit of washout and recovery. And they either got half a ml of the typhoid vaccine or normal saline as the control in their left arm. We were scanning on a Siemens Magnetom Skyro, a 3T scanner with a 32-channel head coil. Took a very standard T1 for segmentation and anatomical reference. This was our 3D EPSI sequence. I'm not going to dwell on this too much, but basically that the metabolite and water reference data sets were obtained with different TRs, and it was manually shimmed to achieve a full with half max of less than 30 hertz across the brain, and we used a sat band across the eyes to suppress signal from the eyes and sinuses, and that's pretty much it. So this scan actually only took 15 minutes, so I lied. So all the data went through the metabolite imaging and data analysis system, or MIDAS software package. So it's a fully automated pipeline including image reconstruction, correction for spatially dependent B0 shifts, spatial registration to MRSI space at 2 millimeters isotropic voxel resolution, and smoothing segmentation to get grey, white and CSF. And then the FIT2 model within MIDAS was used to carry out spectral fitting using Gaussian line shapes, and then it was normalized to institutional units using water as a reference. And then this nifty little formula here to calculate brain temperature in degrees Celsius. So that's the fun thing about this, is that it gives you an absolute brain temperature in a way that you would get if you put a thermometer in your ear or under your arm. So the project review and analysis, or Prana and MINT modules within the MIDAS software were used to analyze the data. So then we used the AAL atlas with 47 regions, and then used a inverse spatial transform to convert AAL regions to subject space. And then the MINT package basically takes all the spectra within a particular region of interest and gets an average to give you one spectrum with really nice SNR. And then we were able to quantify metabolites and calculate brain temperature within each ROI. Here is a quick example of spectra obtained in one region of one participant in their vaccine scan, or the typhoid scan, and then one during their placebo scan. So, very clean, very normal looking spectrum. Here are our results. So all our participants were relatively young. They were mostly based at the university. Equal distribution of males and females, mostly European and Asian, and otherwise very healthy, body weight, body temperature, all within a very normal range. So they were all well. So they didn't have any other other inflammatory things going on the day they came to us. We didn't find anything in terms of the whole brain temperature. So typhoid vaccine didn't appear to affect whole brain temperature, but what we did see was that a significant proportion of the brain regions, so 44 out of the 47 AAL regions, were at a higher temperature post-vaccine than they were post-placebo. And mixed models for the 47 regions showed that a significant treatment on the anterior cingulum, which was a really exciting and I guess expected finding for us, and the left occipital lobe. This is a really yucky picture, but really all I wanted to point out to you was that, so the bars in red are the regions where the temperature went up, and the bars in blue are where they went down. The smallest increase in temperature was 0.01 degrees Celsius in the left mid-cingulum, and the largest was 0.29 degrees Celsius in the right anterior cingulum. So the three regions that didn't increase in temperature post-vaccine was the right frontal lobe, left anterior cingulum, and the left paracentral lobule. So those were our 44 out of 47 brain regions. So looking at the metabolites as well, the significant effects of treatment on the metabolite ratios didn't remain significant after we corrected multiple tests, and there were no significant effects of treatment on the NAA over creatine ratio in any of the 47 brain regions. So we were excited by the study. It was the first one to investigate the effect of an acute inflammatory stimulus on brain temperature and metabolites across the whole brain. And as more than 90% of our 47 brain regions showed an elevated brain temperature post-vaccine. So it shows that we potentially could detect differences in brain temperature, but overall whole brain temperature didn't appear to change. We weren't really that surprised by that in the end, because given that the magnitude of changes in each ROI were really small, so we didn't think that we'd be able to pick that up over the whole brain. And the three regions that decreased in brain temperature included the right frontal lobe, which is a pretty large region on the atlas, and it contributed a lot of voxels to that, so that could have sort of negated any differences that we might have seen in whole brain temperature. And we know, we have seen that whole brain temperatures happen in conditions of really high inflammation, like acute stroke. So in terms of conditions where the inflammation is expected to be lower, like chronic fatigue syndrome or ME/CFS, a study that I did in my postdoc, we didn't see whole brain differences in temperature either, but we did see regional increases in brain temperature. This is pretty much in line with that. Typhoid is a pretty mild acute inflammatory stimulus. So we thought if we were to do this again with a more potent inflammatory stimulus, maybe like lipopolysaccharide, we might see a greater effect on brain temperature. Not sure if ethics will let us do that at this point, but I'd love to. The regions that we did see changes in temperature included a bunch of cortical and subcortical regions, so we do think that the inflammatory response is happening sort of across the brain, but maybe that whole brain temperature isn't really the best measure for it. So that was really cool. We saw what we hoped we would see with the scan, and then I'm just going to take the next two slides to talk about a couple of applications of this scan that we've also been involved in. Oh, I've run out of time. My goodness. Sorry, 2 slides, I promise. So basically they took patients, they put a really cold helmet on them and passed through coolant to try and cool their brain temperature, and this is potentially like a treatment post-stroke, and they were able to shorten the EPSI scan and do it in 8 minutes and do it repeatedly to measure brain temperature, and what they did see was that they a 0.9 degree decrease in brain temperature over the 80 minutes of scanning, which was really fun. And right now, at the moment what we're doing also is applying the EPSI scan to look at cancer-related cognitive impairment, or what we used to call chemobrain or chemofog, because they think that might be related to inflammation. So that's a bit of a watch this space one, because we're still recruiting. So really quick references and the 2 papers of our main brain temperature study and also the cool head study. And thanks to my postdoc, Julia, and the rest of the lab. Sorry.
Thank you, Joanne. Please post your questions in the online Q&A app of OHBM. I'll give the floor to Maria.